Hello everyone, welcome to another video. Today I'll be discussing CSIR NET Life Science, Unit 1, Important Topics and Reference Books. One does not simply learn all the topics of biochemistry the night before the exam. I want you all to understand that this is a very conceptual subject, biochemistry, and just like that, you will not be able to memorize everything. If you talk about the pathways, you might think Krebs cycle you just understand. But the real Krebs cycle is you learn the Krebs cycle, you forget it. You again learn it, you forget it. Repetition is the key. You have many different aspects of this particular unit. You have to understand what is thermodynamics. You have to memorize 20 amino acids. You have to understand enzyme kinetics. You have to know every detail about metabolism that there is to know. This is just few of the aspects. When I'm going to show you the syllabus now, you will understand there is a lot more to it. And how you will do it? I will give you some tips. So stay tuned till the end. Let's look at the weightage of the questions which are asked from Unit 1. These are color coded to most important, which are marked in green, medium important, which are in yellow, and least important, which have not come in exam as of now. Those are marked in blue. The important concepts from here is amino acid, proteins, nucleic acid structure. By important, I mean in every exam, you can expect a question from this. Either a Unit B, that is a Section B, or a Section C question. Stabilizing interactions regarding the different bonds, their properties and especially the bond energy, the bond strength that they have, questions are asked based on that. Thermodynamics, especially the free energy, Gibbs free energy changes, exergonic, endergonic, and application to different processes. Glycolysis, oxidative phosphorylation, in that electron transport chain and ATP synthesis, the different inhibitors which are involved in this process, questions always come from here. Principles of catalysis, enzymology, entire this thing, that is theoretical questions from the concepts, enzymes, kinetics, and enzyme regulation. Again, conformation of protein, Ramachandran plot, earlier they used to ask a question every time in every exam, but of late I haven't seen in 2-3 papers questions on Ramachandran plot. Secondary structure, they are focusing now regarding alpha helix, beta sheet, beta turn. Definitely questions are there from this area. Stability of proteins and nucleic acids, focusing on denaturation, renaturation, and Finson's experiment, the different folding processes, which are the chaperones process pathways involved, and biosynthesis of nucleic acid, nucleotides, the de novo pathway, the regulatory steps, salvage pathway. So these are your most important concepts, but you must not ignore these also, that is the other chemical bonds, covalent bonds, ionic bonds, structure of atoms, because it will give you a foundation, and carbohydrates, lipids. Sometimes questions are there from epimers, anomers, the glycosidic bond, and in lipid, membrane lipids, the adaptation to temperature. Even pH buffer, sometimes you will get question, numerical questions only. Then bioenergetics, coupled reaction, biological energy transducers like NAD+, FADH2, TPP, what are the cofactors, coenzymes, right? Mechanisms of enzyme catalysis, isozymes, till now question hasn't been asked. Reaction kinetics sometimes maybe in 3-4 year one question. Colligative properties haven't come. Vitamins also you may get in 3-4 years one question. But of late they have started giving this microRNA, tRNA. So you must study this part, okay? Because in this exam there were questions on this microRNA and even the RNA stem loop structure. However, conformation of nucleic acid ABZ is also kind of an important topic, but I didn't want to green color code everything. That is why I have color coded this, but consider this as an important topic. Metabolism of carbohydrate vis-a-vis the other pathways like gluconeogenesis, glycogenolysis, pentose phosphate pathway. So these are not that important questions. I haven't seen much, but it is better to study. Lipids, amino acid metabolism, not that important, but amino acid maybe 2-3 years there may be one question. So will be for vitamins also. Okay. Let's jump into the individual units. Must topics to be covered from 1A and 1C. So I will not tell you exclusively from a subunit. I will make combinations. For example, this is from 1A, 1C. which are regarding the bonds, different types of bonds which are involved. What you must study from here, the different properties, occurrence and strength of the types of bonds. Like covalent bond is a strong bond, so its strength is around 110 kilocal per mole, like that. And if you express in joules, it will be 250 kilojoule per mole. Pay attention to units in biochemistry. Okay, this will be important. Ionic bonds are less compared to covalent bond. So what is the basis of the bond? How the bond is formed? You must know that. The stabilizing bonds between macromolecules, example proteins and DNA, like van der Waals hydrogen hydrophobic interaction. What you must pay attention to is that where these bonds are present and where these bond concepts can be applied. Like when you are studying protein folding, the different parts of a protein can be folded together by these stabilizing bonds, or two different subunits of protein can be folded together by these bonds. So there will be complementary binding. This is where you must apply these concepts. Must topics to be covered from Unit 1B, G and I, which is regarding amino acids and proteins. So what is important from here? Questions come as amino acid structure and the behavior according to the different R groups. Properties such as chirality, L amino acid, R amino acid, which is more prevalent, and D amino acids. Then terms such as isoelectric point, pK value is extremely important, because these concepts you will also need in Unit 13 when you are studying the techniques based on them. Then protein secondary structures, as I just said, even the protein function like binding kinetics, cooperativity, what is the binding equilibrium, some formulas based on that, questions might come. Protein conformations, the dihedral angles, phi, chi and omega, Ramachandran plot. Also the techniques to determine different conformations and their sequencing. So this part is more from Unit 13, but combined questions are often seen from this area. Protein folding thermodynamics, denaturation, biological function, whether protein folding is a favorable process or not, what happens to the entropy, what happens to the energy, free energy, these all are important concepts. Must topics covered from this nucleic acid chapters, Unit 1B, 1H and 1J. So the DNA structure, Chargaff's rule, numerical questions come from here. The different forms of DNA, ABZ, their properties, which is right-handed, left-handed, what is the diameter, what is the pitch, rise per base pair, absorbance, denaturation, renaturation, melting point, they will give you some sequences and ask you whose melting point is more. It is based on the GC content. If GC content is more, then it will have a higher temperature. Then we have nucleic acid topology. Not that important, but sometimes you may get a question regarding twist, rid and linking number. Types of RNA, as I said, of late they have started giving question. The tRNA, rRNA, microRNA. This you will also study from Unit 3 because molecular biology will deal with the structures. But focus on what type of sequences and what type of structure it will have. Especially the different forms and variants of the nucleotides that you get in tRNA. Okay, and what are the different arms and what specific type of bases are present over there. Nucleotide biosynthesis from the metabolism, what you should focus on is the regulation and the committed steps. This is what in metabolism overall, wherever you must focus on the regulation part. For example, in purine synthesis, what is the committed step, right? And in pyrimidine synthesis, which is the committed step? Those are the rate limiting steps. What is the activator? What is the inhibitor for that step? These are very important. From enzymology, Unit 1F, enzyme catalysis concepts. All the terms and terminologies like activation energy, coenzyme, then prosthetic groups, right? All these things. Ground state, then transition state. You can get theoretical questions also. In fact, you do get. Okay, enzymes can catalyze to a power of how much? Like 10 to the power 10 to 10 to the power 11 times faster. So just basic properties, based on that also you can get questions. Okay, lock and key model or induced fit model, everything you must know. Conformational changes. Enzyme kinetics, terms and terminologies like Vmax, Km. You will be asked graphical representations of Lineweaver-Burke plot, Eddie-Hofstee plot, Haynes-Wolff plot, and along with the formulas. This is very, very important topic. Definitely one of these questions will be there. The enzyme inhibition, competitive, uncompetitive, mixed, non-competitive, The properties as in the formulas will be different in this case, and also the graphs also will be different. Okay, so each of these different properties along with examples, few examples of those enzymes, like competitive you will find malate and succinate dehydrogenase, so that example you can take. Even in glycolysis also you will find examples. And enzyme regulation, what is allosteric regulation, feedback regulation, what are the rate limiting enzymes that catalyze the rate limiting step, along with examples, very, very important concepts. Must topic covered from subunit 1D, which is regarding pH, buffer and thermodynamics. You will get numerical questions on molarity, pH, buffer, so moles, Dalton, normality, molarity, solution, concentration, percentage, that is weight by weight, weight by volume, volume by volume. These all concepts must be clear with you. Okay. Then numerical questions, enthalpy, entropy and free energy calculation. So formulas have to be crystal clear for you. Delta G is equal to delta H plus T delta S, all these questions, minus T delta S. And in various cellular processes and conceptual questions on thermodynamics, like what is endergonic, exergonic reactions, and especially their application to metabolism. Okay, so everywhere you must think from the point of thermodynamics. For example, in protein folding also, as I mentioned, thermodynamics concept you must know. Focus on formulas. When you study this topic, focus on formulas, unit, log rules, log values also, sometimes log value, anti-log value, and also the power concepts, 10 to the power minus 11, okay, like that. What does it mean? And quick conversions, we must be very pro at. Sometimes graphical questions are also asked from here. For example, if you have a graph like this, then this slope will be representing what? What can you calculate from here? Okay, then this graph, it will represent what? So like that, this graph will represent what? What will be the slope of this graph? So some such kind of questions. But whenever you are getting any graph based questions, you should always see what is the y-axis and x-axis, because that might make you might be making a mistake if you're not looking at it properly. Must topic covered from carbohydrates, lipids and metabolism. So lipid structure with special emphasis on membrane lipids and adaptation to temperature. What happens in low temperature, whether the fatty acids will melt in membrane or not. Then carbohydrate structure, concentrate on epimers, anomers, glycosidic bond formation, types of bond between di and polysaccharides, right? Alpha glycosidic, beta glycosidic, like that. Then glycosaminoglycans, some examples and functions they might ask you. Proteoglycans, these are again common to unit 4, which are regarding the cell communications. So you can have a look at that. Metabolic pathways of carbohydrates including glycolytic and TCA cell cycle. So we have regulation, again important. Focus on the allosteric and feedback inhibition. Then enzyme cofactors involved and also the stoichiometry, means how much ATP is produced. So how much ATP you will get from burning one glucose molecule, complete metabolism, or how much ATP will be produced only from one round of TCA cycle. Also the electron transport chain and ATP synthesis, especially the inhibitors, what inhibitors are there at which step, like for ATP, for electron transport chain, whether they will inhibit complex 1 or complex 3 or complex 4, or ATP synthesis, oligomycin, it will inhibit the ATP synthase enzyme. So these inhibitors are extremely important. You can get some match the following questions also, and even some statement based questions as well. Okay, so I'll be giving you some top concepts like how to prepare unit 1. Master the basics. Okay, most if not all concepts in biochemistry will be difficult to follow if you don't know the basics first. You need to understand the structures and why molecules function the way they do because of their structure. So in this part, the saying that form follows function comes into play. So often this is that it's better to understand first. Okay, the memorization should come second, because most of the names, they are based on their function. So try minimum memorizing. It will be better for you to learn. So before the subject starts, I will suggest that you start brushing up on the chemistry part. Don't have to go thoroughly, read it on top of whatever subject you are studying already. Just familiarize yourself with at least some common terms, functional groups, suffixes which are used. Okay. For pathways, the overall pathway, if it looks very intimidating to you, you can start with small chunks, then link them together. For example, glycolysis is divided into two parts. So you can study the first part, initially preparatory phase, what happens. Then you can study the payoff phase, and then you can link them together like that. Repetition is key. So biochemistry is extremely visual. The more you revise, the more you apply the concepts. You can draw flowcharts or diagrams and look at it more often. It will be helpful. Utilize flashcards and mnemonics. Yes, these are again very important because the more often you will look at the words or the structures or the enzyme names or the regulatory pathways, and you can use some mnemonics to, for example, remember the essential amino acids or maybe the amino acid functional groups. So the more you revise, it will be better. Know your nomenclature. This is a pro tip in which that you might think that scientists will make names out of the blue, okay, just by slapping their hands on a keyboard. But it's not like that. In biochemistry, suffixes will be your best friend because they are reused again and again. The best part is that these are giveaways to their function and substrate too. Some examples like enzymes, they end with A's, right? And then lysis, it would mean that it is to break down. Similarly, O's, it means related to carbohydrate. I just gave some few examples. There are many more like that. And with every step when you study biochemistry, ask yourself why things happen. So with every step, why are some molecules cleaved? Why do we add phosphate groups? Get used to asking why questions, and eventually you will find connections that just click. Okay. So which are the standard reference books? Everyone knows the Bible of biochemistry is Leninger. You can refer one of the latest editions. You can also refer to CSIR. CSIR is also a good book. You can find some few concepts, especially Unit 1C, some detailed concepts in this CSIR book. There is also, for Unit 1C, the bonds, John Kurian, you can refer. That is also a good book. It is available in Google Books. Then Voigt and Voigt, it is a little bit detailed, and I will say a little bit advanced. So until and unless you are pro at biochemistry, you already know the concepts, try to avoid. You can refer CSIR and Leninger. Okay. And some other things, gathers the study material. If you have any ready-made notes from teachers, seniors or friends. The best way is that which you are already prepared, that is Biotecnika premium study material, which are genuine source and they are concise also. So, and most important is the concept notes by Biotecnika. These are ready-made notes, you don't have to worry on that. Everything is color-coded and they are very to the point and important topics are given. So you just have to study and revise from there. Okay. Also, you have the concept wheel books, which gives you a concepts of a particular topic. For example, hydrogen bond, starting from how the bond is forming, what type of specification is there, what is the strength, everything that will be related to hydrogen bond, it is given at one point. So it is very easy to recollect during the exam. Okay. You also have the concept table book, which is mainly on comparison, because in Unit 1 you will need comparison. What is the enzymes name in glycogenolysis and glycogenesis? So how are those enzymes activated? How are they inhibited? For example, here it is glycogen phosphorylase, here it is glycogen synthase. How does glycogen phosphorylase get activated? How does glycogen synthase get activated? When you see side by side, automatically it takes a stamp in your head. So you will never forget these concepts. Conceptica Light, as I said, biochemistry is extremely visual. If you have at one place all the concepts, you can have a look at this book Conceptica Light by Biotecnika. Every concepts will be at one place, and this again, if you look at it time to time, then you don't have to put much effort in memorizing. And the best part is you will not forget that often. Okay, so that is all from here. All the best, everyone. Thank you.